Have you ever wondered how and why we can keep growing taller day by day, and how we can get a new skin after it was torn out, or why we have the same and similar face like our parents? This is all happens because of mitosis and meiosis. But before that, let's get closer to chromosome. Chromosomes are located in nucleus and made by a collapsed chromatin that consists of DNA and genetic material. There is two parts. The short arm is called P arm, while the long arm is called Q arm. If there is just one, it is called chromatin. When there is two like this, it is called sister chromatin and also can be called as chromosome. At the centromere of chromosome, there is not core. Nevertheless, every species of organism have their own fixed number of chromosomes. Such as human, we have 46 chromosomes, and flies they have 4, while cabbage they have 80 chromosomes. Now, let's get into mitosis. Mitosis is a cell division that will divide the parent cell into two daughter cells that will have the same number of chromosomes and also genetic material. It will occur in somatic cell, which is in the animal cell, and also in the meristematic cell, which is the plant cell. This is a cell cycle for the somatic cell and meristematic cell. During G1 and G2, there will be a synthesis of protein, organelle, and also a cell growth. While in S phase, there will be the synthesis of DNA, which is the chromosome will replicate. While in M phase, there is PMAT, prophase, metaphase, anaphase, and telophase. And the last is cytokinesis. Let's start with prophase. In prophase, there will be the attachment of chromatids, and then it will become sister chromatid. And the centrosome will move to opposite poles while organize a magnetic spindle. Fact: there will be no centrioles in meristematic cell, while there is centrioles in somatic cell. Next, the nucleus and nuclear membrane will disappear, and magnetic spindle will attach at the kinetic core of the chromosome. Metaphase. During metaphase, the chromosome will align at the metaphase plate. If there's any problem here, such as the chromosome didn't attach to the magnetic spindle or didn't align at the metaphase plate, the cell will not process to the next process, which is anaphase. Anaphase. During anaphase, each of chromatid will separate and move towards the opposite poles that they face. Each of poles will have the same number of chromatid. Telophase. This is the last stage, which is the nuclear membrane and nucleus will reappear and magnetic spindle will disappear. Lastly, in cytokinesis, for animal cell, the cleavage furrow will form at the center of the cell, in result of cell divided into two daughter cells. Meanwhile, for the plant cell, there will be a cell plate that will separate the cell into two daughter cells. This is a summary of mitosis process in somatic cell. Now, let's check out meiosis. Meiosis is a cell division that happens in gamete cell only, but we have two stages, which is meiosis 1 and meiosis 2, in results of producing 4 haploids of daughter cell. The daughter cell will have a different number of chromosomes and also genetic materials. Meiosis are important in sexual reproduction for gamete cell and also to produce a variation in genetic material among their offspring. For animals, meiosis will occur in testes and ovaries that will produce sperm and also ovum, while at plant will be at anthers and ovaries of flowers that will produce pollen and ovules. This is a cell cycle for gamete cell. The difference here is there will be twice M phase and has interkinesis between meiosis 1 and meiosis 2. Now, let's start with meiosis 1.
During prophase 1,
the homologous chromosome began to pair up in synthesis process, which means the paternal chromosome and the maternal chromosome will pair up. Next, there will be a crossing over between homologous chromosome via chiasmata to exchange DNA. Meanwhile, the centrosomes and centriole will move to the opposite poles, and mitosis spindle will be organized by centrosome. The nuclear membrane and nucleus will disappear before going to metaphase 1. During metaphase 1, the kinetochore of each homologous chromosome will attach at the mitosis spindle while facing the opposite poles, but it is still attached at chiasmata. Now, in anaphase 1, the mitotic spindle will start to pull the homologous chromosome apart to opposite poles, and the chiasmata are broken down but not the cohesin of protein at the sister chromatid of centromere. Now, in telophase 1, the sister chromatid will arrive at the opposite pole. Then, the chromosome will decondense, the nuclear membrane and nucleus will reappear. And now, last, the cytokinesis will occur, which is to separate the cytoplasm into two daughter cells. Before going through meiosis 2, the cell will undergo interkinesis, which is that it will have similar like cell cycle, but they will only have G1 and G2. For meiosis 2, the two daughter cells will divide same as mitosis. So, please go back to mitosis video for explanation. So, before this video ends, this is the comparison of mitosis and meiosis in somatic cell. Thanks for watching. Thank you.